So since it is a neurodegenerative also congress, I will just briefly introduce the disease we are dealing with. It is, of course, Alzheimer's disease, one of the most common neurodegenerative diseases, and the main cause of dementia worldwide. Estimates say that between 60 to 70% of all dementia is caused by Alzheimer's disease pathology. The disease has two main features. One is cognitive decline and memory loss, but initial pathophysiological changes which drive the AD pathology can occur years, if not decades, before the clinical onset of the disease. We also have two important disease hallmarks, which are amyloid beta aggregates and neurofibrillary tangles from tau protein. Luckily, some of those changes can be monitored through cerebrospinal fluid biomarkers such as amyloid beta 42, amyloid beta 42 to 40 ratio, as well as total tau and phosphorylated tau. In fact, decreased amyloid beta as well as increase in tau levels in CSF can also be understood as a AD-related CSF profile in clinical facility. Disease is a multifactorial disease. It also has a complex genetic background, and apart from really rare but highly penetrant mutations in genes involved in processing of amyloid beta, a plethora of other genetic factors can also contribute to the disease susceptibility. Of course, the most well-known risk factor in the late onset type of the disease, which is not associated with those familial cases, is ApoE. But since it's only a tip of an iceberg, a lot of other genetic factors can also play a role in different mechanisms, which can also drive the course of the disease. So yeah, we focus, and I will talk about the late onset Alzheimer's disease, since it contributes to the vast majority of all AD cases. The typical age of onset is around 65 years, and the estimated prevalence in that age group is between 10 to 30%. So with faster aging population, this will also be a major health burden in the future. Disease has a well-known continuum, so from asymptomatic preclinical stage towards more advanced types of the disease. But the intermediate state, which is really important, is the mild cognitive impairment. This is the state where the progressive cognitive impairment is exceeding the one which is expected according to someone's age or educational status. What is known is that all daily living activities are preserved, but not all people with MCI develop Alzheimer's dementia. In fact, 50% of them will develop AD in the next 5 years. But of course, the course of the disease is a bit multifactorial, and the trajectories are different between the different patients. Two other important metabolic processes encompassing the disease are oxidative stress and neuroinflammation. I think we've heard a lot about inflammation today, so I will just briefly introduce some of the oxidative stress and its importance in the Alzheimer's disease. When we talk about oxidative stress, we think about the imbalance between the reactive oxygen species production and the antioxidative mechanisms, which can lead to the damage of the proteins, lipids, and DNA. But also a complex interplay between immune cells in the central nervous system can trigger the neuronal damage and cell loss in advanced stages of the disease. Of course, both of those states have been associated with Alzheimer's disease, with both hallmarks, in fact, so the amyloid beta plaques and neurofibrillary tangles, but also some changes in enzyme activity of the antioxidative mechanisms which are the first line of the defense, have been shown, as well the cytokines levels in different tissues and cells. So yeah, apart from the first step, defense mechanisms which are like enzymatic to prevent further damage from the ROS, we were also a bit interested in the regulation of the oxidative stress, which is under regulation of KEAP1 NRF2 signaling pathway. So some downstream genes can be activated upon those conditions. We aimed to evaluate associations of common polymorphisms in both pathways with cerebrospinal fluid biomarkers, as well as mini-mental state exam scores in patients with MCI and AD. So briefly, we included 54 AD patients in our analysis, together with 14 patients with mild cognitive impairment and a AD-related CSF profile, 20 patients with no changes in biomarker levels were also included, and we also added 62 population as control in the initial stage of assessing the risk of investigated SNPs. We were focusing on 9 genes in the oxidative stress pathways. We tested 16 SNPs of those genes, and additionally 8 SNPs in inflammatory pathways with CASP-PCR. The statistical analysis with cerebrospinal fluid biomarkers as well as cognitive test score was also done. So just take a look at the results. In the AD risk assessment part of our study, we found 2 important correlations of inflammatory genes. So the carriers of polymorphic allele of one of the polymorphisms in TNF gene were associated with lower odds to develop Alzheimer's disease in the entire cohort, and also when the other patients only were included. Additionally, a similar effect was observed for carriers of polymorphic allele in one of the investigated SNPs in interleukin-1 beta gene, but only in the other patients. In terms of oxidative stress, we observed the importance of NF-A2L2, so the gene encoding for NRF nuclear factor, and so differences on both amyloid beta CSF levels as well as total tau have been observed with polymorphic genotypes of different SNPs. Additionally, a increase in amyloid beta ratio has been associated with polymorphic hemoxygenase allele. In inflammatory pathways, the effect of interleukin-1 beta polymorphism was found, which was so the polymorphic allele was associated with higher CSF amyloid beta levels both in the entire cohort as in the AD patients only, and the increase in amyloid beta 42/40 ratio was associated with a gene, with a polymorphism in a gene encoding for a microRNA, which is also involved in the regulation of interleukin production in vitro. So yeah, we also observed numerous associations with mini-mental state examination scores. To sum up and to point out the most important one, so the KEAP1 was associated with, so the polymorphic allele was associated with higher MMSE scores, both in the entire cohort as well as in the AD group only. So we could discuss that those alleles have a protective effect on cognitive decline in our investigated patients. So to sum up and to give it into perspective, we did observe different associations with CSF biomarkers, both amyloid beta and tau pathophysiology, which could point out the additional effect of oxidative stress and inflammatory pathways, in Alzheimer's disease and mild cognitive impairment. And if we add those our results in the broader picture, together with clinical assessment, some functional imaging and blood tests, as well as, of course, well-established CSF biomarkers, we believe we showed that genetic contributions could also be evaluated for earlier and better diagnostics of Alzheimer's disease, dementia, and mild cognitive impairment. So this is it. At the end, I would just like to thank my co-workers from the pharmacogenetics laboratory at University of Ljubljana Faculty of Medicine, especially Katja Goričar, my PhD supervisor, as well as head of our lab, Vita Dolžan, and also a big thanks to Department of Neurology, University Medical Center, led by Milica Gregorić-Kramberger. Thank you.